The brain is a vital part of our body. It's part of our central nervous system, the control center dictating all of our thoughts and actions on a daily basis. But, like all organs, it can malfunction, hindering the transmission of information throughout the network of neurons that keeps us running. That's what happens to patients with ALS. ALS, short for amyotrophic lateral sclerosis, is a neurodegenerative disease in which neurons, specifically ones that control motor function, break down, leading to progressive muscle weakness and other symptoms detrimental to life quality. It's partially caused by a difference in glutamate, a neurotransmitter that regulates memory and learning capabilities in organisms. Now, in terms of finding cures, how exactly should we study diseases like ALS? To experience the research process, we took a type of roundworm called C. elegans, often used by neuroscientists to study brain function due to its similar neuron structure to humans and simplistic nature. We observed 3 different kinds of behaviors in worms: wriggling, thrashing, and pumping, with 3 different worm strains. One strain to mimic the conditions of ALS involving an excess of glutamate, another for worms that were glutamate deficient called EAT4 worms, and lastly, some N2 wild-type worms as control. The overall goal of this experiment is to test the effects of varying glutamate levels on motor functionality. Under a microscope, you can just see the worms squirming around. The wriggling test is pretty simple. We just compare the movement behaviors of each of the worm strains under the microscope focus, and then use a clicker, or a device that manually tracks the number of iterations by the end of the timing period. Here, we basically have a rate of 1 period per left and right movement, and worms move literally like a sinusoidal function, so it's pretty consistent so far. Moving on to the thrashing movement, we begin by setting the micropipette to the correct scale, 500 microliters, which is also half a milliliter. Basically, thrashing behavior only occurs when the worms are actually submerged in water, so we have to pipette a solution called M9 into the plates, which is basically just water and salt mixed together to clean the worms, before viewing the worm's actual behavior. Overall, the results showed that ALS worms thrashed significantly less compared to the wild-type and EAT4 worms, which in general correlated excessive glutamate levels with a degree of disabled motor functionality. Last but not least, we have to examine the worm's behavior with respect to their swallowing capability. Worms prefer to eat a species of bacteria known as E. coli, so here's a plate, and we decided to observe individual worms instead of a whole batch, so we had to use a tool called a worm pick that is made out of platinum. First, we sterilize the pick by using a Bunsen burner and a lighter. Afterwards, I'm able to start picking the worms from one plate to the next, which is always difficult to some extent. The interesting thing is that C. elegans can pump its pharynx up to 300 times a minute, which is characterized by this twitching behavior. After we finished all 3 tests, now came the results and analysis.
Altogether, we found that differing glutamate levels in C. elegans affect their overall motor and pharyngeal functions. In the worms with ALS, much like humans, we see an impairment in overall motor functions. Glutamate production in humans is often associated with learning and memory, while in this scenario, its changes impact the motor functions of our worms. For our ALS worms, we mutated the SOD1 gene, known for having a mutation that causes ALS.
Thank you for watching.
Yay!